guys, today we will be learning about a macromolecule. To be specific, we'll be learning about proteins. If you remember, there are 4 macromolecules, and proteins is one of the ones that AP exams have a lot of questions from. So we really need to understand what's going on with proteins. So with all the macromolecules, you've got to know what elements make them up, because there are multiple questions about it. And you also have to know some of the building blocks and some of those, how do you build them and break them as well. So when it comes to proteins, we see that proteins are made up of C, which is carbon, H, which is hydrogen, O, which is oxygen, N, which is nitrogen. So CHON, okay? And that's very important that you remember proteins have those 4 elements. It's all macromolecules are carbon based. So proteins have CHO, but also N, which is nitrogen. Now, some protein amino acids will have sulfur. So sometimes we say proteins have CHON, and also some will have S, which is sulfur. So there are, when it comes to proteins, you got to understand what their building block is and what that looks like. So the building block of proteins is amino acids. You really got to know what an amino acid looks like. So we'll look at a question where you'll have the structure of amino acid, and I have it on my screen right now. Let's look at it. So here you have an amino acid. There's actually 2 of them here that's going to bond. But as you can see, I'll draw it on the side as well. There is a carbon at the center. Okay, I'll try to draw that. And you have basically, carbon can form these 4 bonds, which is one of the reasons why carbon's so interesting and magical. It has 6 electrons, and in its valence shell, the last shell, it has 4 electrons, which gives it the ability to make these 4 bonds. So carbon's bonded to 4 things here. Let's look at them one at a time. Here, I'll put hydrogen at the top first because that's one of those things. So carbon is bonded to hydrogen. By the way, all these bonds we're going to look at is falling under your covalent bonds, which is what you'll see with macromolecules a lot. But let's look at some other bonds as well, or some other things carbon is bonded to in an amino acid. So carbon also is bonded to amine group, which is NH2. So that's a nitrogen and 2 hydrogens I added right there. So that is an amine group. And it's also, so you have one side of carbon, it's almost like I tend to think of it as NCC, head tail sort of orientation. So on one side you have the amine group NH2. On the other opposite side, the tail side I call it, has carbon double bonded to an oxygen, and also a hydroxide group. So that right there is, the tail end has a carbon, 2 oxygens, and a hydrogen, which is COOH, or also known as carboxyl group. So you got to remember, carbon's bonded to an amine group and a carboxyl group, hydrogen. And here's the last part of it that is super important to understand. The other group that carbon bonds to make an amino acid is the R group. Okay, the R group is where you see all the different varieties of different R groups, 20 of them to be specific. And some of the R groups could be polar, some of them will be non-polar. So it's really important to understand that the polar and non-polar, how they will interact. And also some of them will have charges, positive or negative charges. But really, the property, like how that amino acid will bond and behave and make the protein structure, goes down to its R group a lot. So we will have to understand that as well. The first question here shows you how you can build So you can take two amino acids and join them. And you see this with all macromolecules. You start with a monomer, we call it, so a smaller, like just an amino acid, another one, and when you join them, you see this, general reaction in, that we see with all these macromolecules. We call that dehydration synthesis reaction, where what does dehydration mean? As you join the two, water escapes. Why? Because as you can see, the carboxyl end has a OH and the amine end has the hydrogen, so a H2O escapes, and you have this bond directly between a carbon and a nitrogen that happens, which is known as a peptide bond. So here's a picture. They're actually showing you all of that, the bonding here. Now let's read the question and see what are they trying to do with it. A polypeptide is polymer of amino acids held together by peptide bonds. So they gave us all that info. The process of dehydration synthesis creates these peptide bonds, and they're showing a picture of it. As you can see in the picture, an amino acid must have which of the following properties in order to be incorporated into a polypeptide? And it's also telling us in this picture, amino acids are linked through the formation of peptide bonds. Let's look at the choices then, and see if we can figure out which one's the correct one. So the first choice here is talking about the ability to remain stable in the presence of water molecules. That's not a property really that explains this at all, like ability to remain stable in the presence of water molecules. I don't know. There must be a better one in this. The ability to form a covalent bond with its NH2 group and its COOH group, amine group and carboxyl group. That sounds like exactly what is happening over here. You saw the amino end and the carboxyl end losing its hydrogen and hydroxide for it to form the bond, which we call the peptide bond. So I would choose B for this particular question, because that seems to answer it. Now, if you keep reading, C says an R group that is compatible with the R group of the last amino acid incorporated. That doesn't explain it at all, because the R groups have nothing to do with taking an amino acid and bonding with another one. R groups, we look at the proteins having this processing to be, going through primary, secondary, tertiary, quaternary, like structural processing for it to be a functional protein, and we'll see R groups have something to do with that. But right now, it, just to bond between one amino acid and another one, R group really isn't doing anything. D says a central carbon atom that reacts with a nitrogen atom to form the peptide bond. Again, the bond here is between a nitrogen and a carbon from the carboxyl and amine group. It is not happening with the central carbon, so that's not true. So that's the kind of questions you will expect from macromolecules like protein. Here's another one. Which of the following is common feature of the illustrated reactions showing the linking of monomers to form macromolecules? So here they have this reaction shown. They actually have two different ones. On top they have sugar. I know that it's sugar from the chemical structure. A monosaccharide, another monosaccharide, going through the same dehydration synthesis reaction to form a disaccharide. And the second one is showing you the NCC tells me this is an amino acid bonding with another amino acid. And again, water is escaping the reaction from both of them. And you are forming this peptide bond and this, protein right there. So two of them reactions showing the linking of monomers to form macromolecules. Right there. So now let's read the choices, because it wants to know which of the following is common feature. Okay? All right. A says two identical monomers are joined by a covalent bond. Identical monomers is where I question, because as you can see from the protein one right here, if you look at the R group here, one has CH3, one has H, so they are different amino acids, so they're not identical. Monomers are joined by ionic bonds, and we know monomers are not joined by ionic bonds, so I'm going to move on. Two different monomers are joined by a covalent bond. That does explain it. It is two different monomers and it is joined by a covalent bond. So I would pick that, but let's go on and read D. Monomers are joined by a covalent bond and water molecule is produced. Ooh, this one really hits it. It's talking about monomers are joined by a covalent bond, which is also what C talks about. But now it's also saying it's describing the dehydration synthesis reaction. So even though C seemed like, oh, that's true, they're joined by covalent bonds, it didn't tell us the whole story. D did though. They're joined by covalent bond, but also water molecule is produced because that's a dehydration synthesis reaction. So for 14, D would really be the right answer. I am going to solve another amino acid question here. This one's interesting. This one is a data question, like they gave you a graph here, and you would think this has nothing to do with understanding proteins, but it actually does have something to do with it. So let's go ahead and read it. The figure shows the results of an experiment to investigate the effects of an enriched CO2 environment on plant growth. Identical plants were separated into different groups and grown either on a standard CO2 environment with 400 ppm CO2 or in an enriched CO2 environment. So really the plants they're talking about, they were pretty much, they're saying it was identical plants, but they were separated into groups. One had a lot of CO2, like 400 ppm, which is the standard level, and the other one has extra, 700 ppm, which is enriched CO2. Of the plants in each environment, half were grown under ideal conditions and half were grown under stressed conditions. Okay, and that's the data we're looking at. Based on the figure, which statement best describes the observed relationship between atmospheric CO2 enrichment and plant growth under ideal and stressed conditions? Okay, so as you know, plants are carbon sinks. They, through photosynthesis, they will take in the CO2, and that goes into building their macromolecules. And that's where you see a plant almost grow out of nothing. They're taking water and CO2 and growing. So it does have to do with macromolecules. But in this question, the data is already given, and we need to just figure out which one of them. They're trying to see if you can read data like this, really. So that's really what's going on with a question like this. So let's read them and we'll see which one's true. The increase in atmospheric CO2 resulted in a greater increase in plant growth under ideal conditions than under stress conditions. So let's actually look at the data one more time. It seems like the increased amount of CO2 right here, this is the darker color, the black bar over here, it is higher dry weight, so more growth. And it's also showing that between, if you compare the ideal and the stressed conditions, the ideal conditions did grow more than the stressed ones. So does that go with it? It says increase in atmospheric CO2 resulted in a greater increase. So far, yes, in plant growth. And is it true it's ideal conditions? It is. So it does seem like A might be it, but let's keep reading it. The increase in atmospheric CO2 resulted in a greater increase in plant growth, again similar to A, under stressed conditions. So now that cannot be true. Increase in atmospheric CO2, and this is C, had no observable effect. Also cannot be true. It's quite dramatic, the effect in here. Increase in atmospheric CO2 resulted in an inhibition of plant growth. Also not true. So really A was the right answer for this one. One more protein related question. This one also shows how they check your understanding on mutation a little bit, but also ties this down to macromolecules. You'll see a lot of mutation questions in AP exam a lot of times, so it's really good to understand the effect. um, Here it says figure 1 shows 3 amino acids, okay, and we can see that figure 1 has 3 amino acid: aspartic acid, cysteine, and lysine, so 3 of them. 3 amino acids that are part of a polypeptide chain. Figure 2, on the other hand, which is this right here, shows the same section of the chain after a mutation. If you read it, this one has aspartic acid, cysteine, lysine in 1. 2 has aspartic acid, valine, and lysine. So val is the second amino acid, is where something happened. So from cysteine, it somehow became valine. So there's a problem right there. Now, how might this change affect the structure and function of the protein? That is the question. So they gave the answer choices, and we're going to go through them to see which one is going to be correct. Since cysteine and valine is a pretty big change here, you're going from one amino acid to another one, there's probably something going to happen with protein function. Let's see which one makes sense though. The new amino acid, valine, has replaced cysteine, okay, which is true. We see that in the mutation in the new protein. Since the number of amino acids has remained the same, there will be no change in the 3D folding or function of the protein. Now, we can't say that, even though the number is the same, because you switch the amino acids and switch the R groups from the amino acids. We know it can affect a lot of things in protein function, because the R groups, they can differ. They might be polar or non-polar, charged or not. So really we can't say that for sure. B says since this is a linear section, it does not influence protein folding. How does that have to do with protein folding? Like, even though you have a polypeptide chain starting with primary sequence that could be linear or whatnot, you will go through processing, it will fold and all, and it will affect others too. So that also doesn't make sense. C says since the new amino acid is bounded on one side by an amino acid with a negatively charged R group, and by an amino acid on the other side with a positively charged R group, the charges will balance and the protein will fold as usual. Not true. In fact, you don't see that in your amino acid R group at all. So I'm going to move on to D. Hopefully that's the answer. The R group of the new amino acid valine has different chemical properties than the R group of cysteine. This will cause the protein to misfold and not function properly in the cell. The misfold part is important to understand, because it is true when it comes to those amino acids, the way they fold and they bond with itself has to do with those R groups. You'll see a lot of times the hydrogen bonds can happen between different R groups as well. So this one does make the most sense, so I am going to pick D. Last MCQ from our proteins is talking about which of the following describes a key difference among the 20 amino acids that are used to make proteins. Okay, this is a pretty simple question, so let's look at the choices. Only some amino acids have a carboxyl group. That's not true. All amino acids have a carboxyl group. Some amino acids contain the element phosphorus. Also, if you think about it, with the macromolecules, carbs are CHO, lipids are CHO, proteins are CHON, and nucleic acids are CHONP. And that doesn't sound like that's true then, because this is talking about protein and not nucleic acids. So moving on to C. Only some amino acids have an R group. That's also not true. We just saw the structure of an amino acid and they all have an R group. So it has to be D, and it is D. Some amino acids are hydrophobic. That does describe it. We just talked about that some amino acids, the R group, will be polar, some will be non-polar. So that means some are hydrophilic, some are hydrophobic. So that does really target a key difference among the 20 amino acids. And that is the right answer choice. All right, that's all about proteins today. Next, we will learn about another macromolecule.